But we'll get started. So welcome to University of Connecticut and University of California, San Francisco's Brainlands Lab meeting. For those of you who do not know me and saw this advertisement on Facebook or Twitter or LinkedIn, I am Maaike Vandermosten, Professor of Psychological Sciences, Mathematics, Neuroscience, and Psychiatry, and Director of the Brain Imaging Research Center at University of Connecticut, and Adjunct Professor of Psychiatry at University of California, San Francisco. So today is our great pleasure to introduce Professor Maaike Vandermosten. I tend to be long on the introduction, so I'm going to try to keep it as short as possible. She has worked in our lab at UCSF in 2015. It was great to have her and her family in San Francisco. Maaike is a developmental cognitive neuroscientist and tenure-track professor in neurological language disorders in the Department of Neuroscience at KU Leuven in Belgium. She has a master's degree in speech pathology and audiology sciences and a PhD in educational sciences from KU Leuven. Her expertise is broad and spans from the developmental neuroimaging, in particular neuroconnectivity approaches such as diffusion MRI, but also in functional MRI and using approaches such as machine learning. She's also an expert in reading, speech, and auditory processing, and also in developmental language disorders such as dyslexia. But more recently, since she took on this, her faculty position in acquired language disorders as well, such as aphasia. And she's been a pioneer in all of these areas. Her work on white matter organization as it relates to reading and dyslexia, as well as work on phonetic representation in children with and without risk for dyslexia, among many of her other work she has done, have influenced all of us in the field. She's certainly my go-to person when it comes to diffusion MRI, auditory processing, family history impact, and offspring's neural and cognitive processes. She continues to publish extensively despite having two young, amazing children, which I find quite remarkable, and in high-impact journals such as Science, PNAS, Neuroscience and Biobehavioral Reviews, and Brain. She has won many awards, but most notably the Rebecca Sandak Young Investigator Award in Reading Research from the Society for the Scientific Studies of Reading in 2016. Please join me in welcoming her. I'm going to unmute everyone. Or Matt, can you try to unmute everyone? I'm going to try. All right. So now, if you'd like to, please do clap. Dogs, children, welcome as well, but I'm going to mute you, everyone, back. And unmute the speaker, Maaike. I can't.
All right. Okay, so I think you can all hear me. All right. Okay, so thanks a lot for the very kind introduction, Fumiko. I look forward to give this talk, and it's also very nice to see some familiar names. Although we cannot meet live, at least we can meet virtually. So, I'm looking forward to the feedback from all of you. So today I will talk about brain plasticity during typical and atypical reading development. I will mainly focus on structural MRI because this is, we have more longitudinal structural MRI, so I will talk mainly about structural changes throughout reading development. Of course, this research is sorry, I think my slides are not, all right. So this research is of course a result of combined efforts of many researchers, and we are grouped within the Dyslexia collaboration team, so the DISCO team at KU Leuven, and I'm coordinating the MRI parts, and this will also be the part that I will present today. So there's also behavioral and EEG data in many of the studies that I will present, but my focus will be on the structural MRI. Okay, so before I start, I would just give like a very short summary about how, what's the neural basis of reading and dyslexia. This is of course a bit of a simplification, but just to give an idea. And the picture that emerges is mainly from matured readers. So based on fMRI studies, what is generally known is that if you read, you activate some regions, some dorsal regions, such as angular gyrus, SMG, and STG, together with IFG, and that supports reading by decoding. So if you really learn by, read the word by grapheme phoneme coupling, you also activate ventral regions, so the ventral occipital temporal region, which hosts the visual word from area, and these regions are more active when you read by direct word recognition. With regard to dyslexia, there was a meta-analysis of Fabio that showed that there is some differences between the activation pattern of dyslexic readers and typical readers, and that the dyslexic readers mainly have a hypoactivation in the left posterior regions. There's also some overactivation in some frontal regions and a bit also in the right, but that was a bit less consistent. With regard to structural MRI, we looked in our own this co-collaboration, we looked at the white matter tracts that could support reading. So for the dorsal stream, we looked at the arcuate fasciculus, which you see in the upper part of the figure. And we also looked at the ventral regions, the connection between the ventral regions, which is through the IFOF. And we saw that, that these dorsal tract, so the arcuate fasciculus is mainly sustaining phonological aspects of reading, and the ventral IFOF is sustaining more orthographic aspects of reading. We also found that adults with dyslexia, they have decreased FA in the left arcuate fasciculus, mainly in the direct or the long segments, so which you see in green, and they didn't have any other deficits in the right or in the ventral regions. Also, other studies, this is a meta-analysis based on VBM, showed also reduced gray matter in bilateral temporal parietal regions. Also, another meta-analysis found is that with VBM studies, you also see some differences between dyslexics and typical readers. If you look at the overlap between the functional studies and these VBM studies, you see that there is especially a deficit in the left fusiform and the left SMG. So this is a bit the general picture in mature readers, how the neural basis is in these mature readers. But of course, we're interested in reading development, and this is something which is very dynamic. A lot of changes occur, but they occur early in development, so not during the periods where we have most of the MRI scans. So if you look at reading development, there's a lot of things changing, and it starts already in kindergarten when children they start to have some awareness of syllables and later of the phonemes, and they start to identify letters. Then when they go to grade 1, they really learn to read. This is really this grapheme phoneme coupling that they learn, so really decoding a word. And during the first years of primary school, there's a lot of focus on this grapheme phoneme coupling. In the later stages of primary school, you see that there is a lot of increase in the speed of reading, and then the focus is maybe a bit more to text comprehension. So you read to learn instead of learning to read. So, but there's still a lot of changes ongoing at you see here, for example, this is from a Dutch study. So with Dutch-speaking children, as most of our studies also have Dutch-speaking children. And you see in these first years, so the orange line is grade 3. So you see that by grade 3, there's the accuracy of reading almost is at ceiling effect. So there is in the first years a lot of increase in the accuracy, but you see in the later stages, so from grade 3 to grade 6, there is still a lot of increase in the efficiency. So in how fast you can identify the word. So later on, with regard to more the technical aspects of reading, so not reading comprehension, you see there is more stabilization. But as I said, the picture I showed you about the neural basis of reading, that's then often in these older subjects, so in adults and in older school-age children. So this indeed poses a question. So what we saw in these previous studies, is that something, what can we conclude from it? Because there is also a very influential theory by De Haan that says like our brain is not pre-wired for reading, so that reading is a relative recent cultural invention, so that we don't have a brain that is made for reading, and that throughout the course of reading development, you need to reorganize already existing networks, such as the language network, the visual network, but so that it's not something that is already present. So therefore, I'm very much interested in seeing how the brain changes during reading development, because I think if we look at an adult brain for reading, then we have a few problems. First of all, if we look at the adult brain and we see some differences related to dyslexia, they might be a consequence of years of reading failure, and they might not be the cause. So therefore, it's important to look pre-reading to see whether we can find some early markers of dyslexia. And this will also help us in finding early predictors of dyslexia, so that we already at an early age can see whether we can predict who will develop dyslexia. So this is one aspect I will talk about. Second, I think it's very important to look at changes in the brain while you learn to read and write. So really across the primary school, see how the brain is changing, because reading development is also a very dynamic process. So preferably you want longitudinal studies to investigate that, because that's these type of studies, they are sensitive to these developmental changes much more than cross-sectional studies. And then a third aspect I will talk about is to see if we really want to see how the brain is changing due to learning to read and write. So whether it's whether this reading induces some brain changes, then I think we need to go to intervention studies to disentangle these reading-induced changes from maturational changes. So I will also talk a bit about intervention studies. And then lastly, I will also talk a bit about how the child is, how the family context also explains a lot of the brain development in children. Okay, so sorry, I'm just, okay. So what I will do for each of these four aspects is first give a very brief overview of the previous studies that have been done on that aspect, and then present some of our own results. But I will also discuss a bit the methodological challenges we encountered and how we dealt with it, because it's very inherent to longitudinal studies in pediatric samples. So with regard to the first aspect, MRI pre-reading markers, if we look at previous studies, first of all, if we just look at the comparison between the activation patterns for reading in adults versus children, there is a lot of overlap, but you also see some differences, which is, for example, shown in this meta-analysis. Also with regard to dyslexia, if you compare children with dyslexia and adults with dyslexia, there is some overlap in the location of the deficits, but there is also some differences. So that showed a bit that there is a need to also to look at an early age and not to wait until they're already matured readers. And so there was a lot of a new tendency was to indeed look with MRI to pre-reading children. So this is, for example, also a review that we did based on the available studies back then. But of course, so we did see some differences between at-risk children and children who were not at risk for dyslexia. But of course, you have to keep in mind that at that time, there was not yet a classification of who developed dyslexia and who didn't. So it was based on a risk for dyslexia and not on the actual reading problems they encountered. We also, in this review, we really put all the studies together, regardless of what the MRI modality was. And also, there was no longitudinal follow-up in these studies with the MRI. So that led us to investigate these pre-reading MRI markers in a project that we started here at KU Leuven. What we did is we started with a group of pre-reading children, so in kindergarten, and half of them had a family risk, half of them did not have a family risk. And this was done in order to increase the number of children who eventually would develop dyslexia. And so then these children were in kindergarten. We did some behavioral assessment on the typical reading precursors like phonological awareness, rapid automatic naming, letter knowledge. But we also did an MRI scan in 72 of them. And then we followed them up throughout primary school. So we had every 2 years an MRI scan, and every year we had behavioral data that we collected. Given that we had also reading data, of course, throughout primary school, we could classify who of our included children had severe and persistent reading difficulties, so who would be classified as dyslexic and who was classified as typical readers. So at the end, we had around 15 dyslexic readers in our MRI data and 40 typical readers. The exact numbers vary a bit depending on which modality was used, so, but this is more or less the number of dyslexic and typical readers. So with regard to the first aspect, so looking at pre-reading markers, so I will mainly discuss the MRI data that were collected at this pre-reading stage. So if we first have a look at the white matter, what did we find? So we looked, so this was work of Leuven van der Oude, so the work she did within her PhD. And so within that project, we looked at a typical reading-related white matter tract, so the dorsal arcuate fasciculus with its 3 segments, and the ventral IFOF. With regard to the method, we did all analysis in native space, so we did a manual tracing of the white matter tracts. So it's for each subject we did it, and this avoided some problems with normalization, which you can have if you normalize children data to an adult templates, for example. But of course, it also has a disadvantage that it's time intensive, and there's also some inter-rater variability, but this was quite okay, the inter-rater variability in our sample, because we checked it with 2 raters. So what were the results? In this pre-reading stage, so we saw that there was a difference between children who eventually developed typical reading skills and children who developed dyslexia. Sorry, I see there is a typo in the title here. So so you see the NR is for normal readers and the DR is for dyslexic readers. But so with regard to group differences, we saw that there was a decreased FA in the children, in the pre-reading children with dyslexia in the left arcuate fasciculus, which is in line with the findings we had in the adults, but we also saw it in the right arcuate fasciculus. So again, in this long segment, so now we found different than in the adults, we found bilateral differences between the groups. And then we also did some logistic regression to see, can we predict who will develop dyslexia, yes or no? Of course, you have to know that given that 75% of our group was typical reader, even without including anything in the model, we can classify 75% correct as typical readers. So everything above 75 is an increase in the prediction. And we saw when we entered the white matter predictors, we could increase indeed the correct classification to 85%. And it was mainly the left arcuate fasciculus, which was a significant predictor, and so not the right. And this left arcuate fasciculus had a unique prediction on top of the typical behavioral predictors. We also had a look at correlations with behavioral, just to see in the adults, we saw that there was some kind of dichotomy, that the dorsal tract was more related to phonological stuff and the ventral IFOF more to orthographic processing. In this pre-reading, we saw that there was indeed this correlation with the left arcuate fasciculus and phonological awareness. But phonological awareness was also related to the bilateral IFOF, which was not the case in the adults. So there seems to be less of a neural specialization, dorsal ventral presence. And then we also looked at the T1 images, so at the gray matter structure. And this was part of the PhD of Caroline Belen. So we decided to look at regions that were potentially interesting for dyslexia based on the meta-analysis on MRI studies in dyslexia. And we used free surfer analysis to investigate gray matter volume, thickness, and surface area in these regions. I will talk a bit more about the method later, but I'll first present some of the results. So when we compared typical readers with dyslexic readers at the pre-reading age, we saw there was some differences in the surface area and also in the volume. In the left fusiform gyrus, which was probably expected because it hosts the visual word form area, but we also saw it in the right. So again, as with the diffusion data, we saw more bilateral deficits than we observed in adults. In the thickness, we did not see any differences. We also complemented the region of interest analysis with a whole brain approach, and there we saw it confirmed the presence of of a decreased volume in the left fusiform gyrus. The right was not found, only when we had a more liberal thresholds. So these were the findings in the gray matter. So if and then with regard to the correlations with the behavioral measures, we again saw that these phonological skills correlated with ventral regions like the fusiform. So again, this confirmed a bit that at the pre-reading age, also ventral regions are related to phonological processing. So, and then a bit more about the methods, about the free surfer method we used for the gray matter volume. So as I said, we use free surfer. So we used it because it's automated. But because it was children data, there are more, the quality of the data is less. And also the difference between the children data and the adult template that we used within the free surfer analysis, there's a bigger differences. So so we did manual edits for each of the images of the children. So it's around 5 hours per subject. So it was very time intensive. So afterwards, we wanted to see, was it worth the effort? So we compared our results, the morphological outcomes with manual edits relative to without manual edits. And we see indeed in absolute values, you have differences in the volumes between with edits and without manual edits. But overall, it was consistent across subjects. So there was a high interclass correlation between the methods with and without manual edits. So I think as long as you do, within one study, you should not do part of the group with edits and part without, because then you will get some strange results. But overall, so it results, doing the manual edits results in some differences, but it's highly consistent across the subjects. And also our results with regard to the fusiform was also confirmed if you used the methods without manual edits. So we didn't see a lot of added value of using the manual edits. We did also investigate a bit more, and that was within the PhD of Tan Van Pham, whether the segmentation method that is used within free surfer, which is surface-based, and the fact that we used an adult atlas within this free surfer analysis, whether this has maybe, maybe this is a disadvantage. And so we compared it relative to a method that we developed where we used an age-met atlas, and we used a different type of segmentation, so probabilistic segmentation method, and that was called child metrics. And we saw that when we used this child metrics segmentation and extraction of the volume, we saw there was an increased reliability relative to free surfer, also more robustness against motion, and also a higher accuracy when we compared it with manual segmentations, and there was also a higher sensitivity to group differences. So this indicates that maybe the free surfer method we used was not ideal for pre-reading data. However, when we looked at the clinical findings we found at the group differences in the fusiform, we also confirmed it with this new or this other method. So this was showing a bit of the robustness of this finding. So even with a different segmentation and a different atlas, we still found the differences in the fusiform. And I think, with regard to the future, if you have pre-reading data, whether you can use free surfer or not, I think the new development of the fast surfer is probably very promising, because it has been shown to be like more reliable and more accurate. So I think in the future, this is definitely something to try out, but it's, of course, it needs to be checked whether it's applicable to, or whether it's working well in pre-reading data. Okay, and then, so in the previous slides, I mainly presented some results where I compared dyslexic readers with typical readers. But of course, we also compared family risk without family risk children to children without a family risk, because this is how they were originally recruited. And so there we saw that we saw some temporal regions where these two family risk groups were different. We observed it with the gray matter volume, but we also observed it in the FA of the left IFOF and right IFOF as well. So also in these temporal regions, and also with an fMRI study. I will not go into detail about that study, but we looked at how distinct the neural representations were in this between family risk and between non-family risk children, and we also saw some group differences there. So it seems that this temporal region seems to be like a risk factor for dyslexia, but it doesn't really relate to the reading skills they eventually develop. Okay, some interim conclusion about these pre-reading markers. So we did observe some early deficit in the left reading network. So we saw it in the left arcuate fasciculus, and this was also confirmed in later studies that also used pre-reading samples. And also with regard to the left fusiform, this was also replicated in another sample. So it seems to be that indeed already pre-reading you have some deficits in this left reading network. So it's not purely, so the deficits we observed in the adults are not just a consequence of the reading failure they had years and years, but it might be at the basis of their reading problems. Though we did not see yet this in terms of functional specialization of the dorsal and the ventral stream at the pre-reading age. We did not see that yet. And also what's different with the adults, we really saw deficits in left and right. So not only in the left hemisphere. This might seem a bit in contrast with like the recent proposals that maybe the right hemisphere is like a protective factor for dyslexia, because in the study of Zook, they, for example, demonstrated that within the group of at-risk children, the ones that develop typical reading skills, they have a higher FA in the rights. But of course, it might still be that our results are not in contrast with these results. But I think what we need to do is to investigate a much larger sample of children at risk and then see the ones who develop typical reading skills and the ones who don't develop reading skills, whether they differ in the right hemisphere. And so this is also what Leuven will be doing, is doing already for the past year, but also for the next years. So she's recruiting extra children, pre-reading children with a cognitive risk. And so the idea is to merge the new sample with the sample that I presented. And this will also help in disentangle a bit more what's now the role of the family risk, because it seems, at least on the data we currently have, that there is some risk in the temporal lobe, but it does not necessarily lead to poor reading outcomes. Okay, so this was about the pre-reading markers. We also, of course, looked at the plasticity during reading development, typical and atypical readers. So it's the same, so just to tell you a bit more about previous studies, I'll start with fMRI studies, because these studies, as I said, the neural recycle theory of de Haan is mainly based on fMRI studies. So the idea is that before you start to read and write, the visual word form area, so which is in the fusiform gyrus, is not yet specialized for recognizing words, but once you learn to read and write, there is some increased specialization, and this has been confirmed now also in longitudinal studies. Also in the more dorsal regions, you see some changes ongoing when you start to read and write. But the question is now, many of these studies, of the fMRI studies, really focus on this early reading stage. So there's not a lot of information across the whole primary school. And also, it's also not known whether these functional changes, is it also reflected in structural changes across the reading development. So if we look at the studies that are available on T1, so on the gray matter volume, we see that, so these are mainly large-scale studies, but yeah, within the range of primary school, there's not so much data available. So especially these first grades are often underrepresented in these large-scale studies. Also, we see that at the tissue level, it seems that the gray matter volume is rather stable, so not decreasing or increasing in volume. But we don't know at a sub-regional level. So of course, you can have some regions that are still increasing and others that are decreasing during specific stages of primary school. And we believe that this information is very relevant, because if you have regions that co-develop during a certain period of primary school, that might indicate that they form some kind of network. And so this information is very relevant to know which regions co-develop during specific stages of primary school. And also there's not a lot of studies that really relate these changes to reading development. And also that investigated whether dyslexic then maybe have an atypical reading trajectory, which is reflected also by an atypical neurotrajectory. With regard to diffusion studies, so mainly looking at the white matter, we see that in contrast to gray matter, there is more consensus that there across primary school there is an increase in FA or in the volume. So it means that there is a steadily increase in white matter. If we look at studies that looked at really at the relation with reading development, we see that there are some studies that show, for example, that the more the temporoparietal region is increasing white matter volume, the better reader you are. So it seems to suggest that, okay, if you're a good reader, you need to have a lot of plasticity in the white matter. And that was also in another study of the group of Gab, so of Wang, showed also that the rate of FA development is lower in poor readers than in good readers. So this seems to indicate that there's maybe different neurotrajectories, at least for the Arcuate fasciculus, in poor versus typical readers. But of course, there's not so much information about these specific reading stages during primary school, and also not about children who eventually develop dyslexia, because in these studies often it's about a typical range of reading skills or poor readers, but not really children who have persistent and severe difficulties. So therefore, we had the current project, so it's the same data as I presented for the pre-reading markers, but now we will focus a bit more about the changes that happened between the different scans, so between pre-reading and grade 2, and also between grade 2 and grade 5. So if we look at the diffusion data, so at the white matter, first of all, with regard to the relation with behavioral measures, we see that, as I said, pre-reading, there was not yet a specialization, so also the ventral regions were sustaining these phonological skills. We see by grade 2, there is this dorsal ventral specialization with dorsal regions maybe, sustaining phonological skills and ventral regions sustaining orthographic skills. But then with regard to, if we really want to look at this plasticity across the whole primary school, so across the 3 time points, we changed a bit in methods. So the previous diffusion MRI data were all with manual tracing, and now we switched it a bit to a more automated approach. There were 3 regions because it's very time intensive to analyze all the 3 waves with manual tracing, but also because the study of Wang used also this automated approach, and it allows to look more within the tract at which part of the tract is now different between dyslexics and typical readers. So it gives you a bit more information about where in the tract you can find some differences. Although it's still, I want to emphasize that even with this automated method, it's still in native space, the fiber tracking. It's just that the regions of interest that you use for this fiber tracking, these are based on an adult's brain. And so then they are projected towards the native space of the child. So I think with regard to pediatric data, it's still okay because you still do the fiber tracking in native space. So if we look at the results, we saw that the pre-reading deficits within the tract were mainly within nodes 90 to 22. And we saw that these group differences, that they remained present over the different time periods. So they were present pre-reading, but also still early reading and in the advanced reading periods. And there was no interaction with time. So this seems to suggest that there is between typical and dyslexic readers, that they have a similar neurotrajectory. So, which is in contrast to what the findings were of the study of Wang, because they found different rates of FA development in poor versus typical readers. Okay, so that was for the white matter. If we look at the gray matter, I will first say something about the method, because I think with regard to the T1 data, the traditional approach is to integrate, to just analyze the T1 data at the first wave, and then you analyze them at the second wave, and you integrate them in the statistics. But there is a more recent approach, and this is also the approach used in FreeSurfer, is to really integrate already at the image level, because this would result to a higher reliability. The only problem is we couldn't use the longitudinal pipeline of FreeSurfer, because FreeSurfer assumes that the head size stays stable across the different waves. And we saw that here, if you look at the stage, at the early reading stage, so from pre-reading to grade 2, there was a significant head size growth, and also in the later stages, so from grade 2 to grade 5. So therefore we adjusted, or we developed a method that also integrated the T1 data at the image level. So we did, again, like this within subject time point registration, as is done with FreeSurfer, but now we took into account the head size growth. So I will not go into detail, but the idea is that at the end, you have a measure that gives you an idea of the percentage volume change, but it takes into account the head size growth. So it was also, we showed that it was also more reliable than if we would get a measure of this change in a cross-sectional way. So I think this is important to know. So what were the results? So first of all, maybe in order to interpret the changes we see so we have a measure of gray matter change. In order to interpret them, I think it's important to say a bit what we expect to see in terms of maturation, of gray matter maturation, at least at the macroscopic level. So the level we measure with MRI, we expect to see an inverted U shape. So because in the beginning, at the microscopic level, you have a lot of extrasynaptic connections that are made, and also some extra myelination, and this would result in a positive PBVC, so in a positive, so in an increasing gray matter change. Then later on, we expect that there is a plateau, so where there you see no change. And then in a final stage of maturation, you would see a decreasing gray matter volume, so a negative PBVC due to synaptic pruning. So if we look within our sample, and we first look at the change between pre-reading to early reading, pre-reading to grade 2, so the early reading stage, we saw, first of all, at the tissue level that gray matter was stable, but we did see that at the regional level, there's a lot of differences. So some regions increase and some regions decrease in gray matter volume. The increase was mainly seen in regions involved in the left, in reading, so part of the left reading network. So there, they seem to be in an early stage of maturation, whereas occipital regions were more decreasing. So they were already in a later stage of maturation. If we look then at the later years of primary school, so between grade 2 and grade 5, we see again gray matter at the tissue level, it's stable, but again we see regional differences with frontal regions now increasing, so being more in early stage of maturation, and then a lot of other regions decreasing, including again the occipital regions, but also now parietal regions are decreasing. So with regard to dyslexia, do they have the same neurotrajectories as the typical readers? In the diffusion data, we did not find a lot of evidence for different neurotrajectories. In the gray matter, it was the same within the reading network. The trajectories were the same between typical and dyslexic readers, but we did see in the right IFG, in the pars opercularis, that their dyslexic readers had more of an increase in the volume than typical readers did, and also in the later stages of reading, so in the later years of primary schools, we saw that both groups decreased in volume in the right cingulate, but that the dyslexic readers decreased more than the typical readers. So some interim conclusions. So the typical, about the typical neurotrajectories, we see there is an FA increase in white matter tracts across primary schools. So With regard to white matter, it's more like a linear increase. With regard to gray matter, we see it's very region dependent. And if we look at the regions that showed an increase, so that were more at this early stage of maturation, we saw them mainly in the left reading network during this early or during the first years of primary schools, and during the later stages of reading, so the later years of primary school, we saw more increases in the frontal regions. And so given that the plasticity of the reading regions is mainly during this early stage, this could indicate that we should maybe expect more plasticity during these early stages of reading developments. And so this is a bit a dyslexia paradox, because in practice, children with dyslexia are often only go for therapy once they are after grade 3. So then probably the brain is a bit less plastic. With regard to dyslexia, we did not find a lot of evidence for atypical neurotrajectories with the left reading network. This is different than what Wang found, for example, with diffusion data as well, but we have 3 data points, so maybe that's a bit more reliable than 2 data points, but of course this needs further investigation. We did see some differences in the trajectories outside the reading network, so this could be some kind of compensation. Okay, I see I'm running a bit out of time, so I'll try to speed up a bit so that I'm not taking all of your day or all of the evening.
No, you have time. Don't worry. I think we had planned, usually we plan for 1 hour plus for these things. So feel free to go at your own pace. Thank you.
Then I keep on going.
Yeah.
Okay, but interrupt me if I'm taking too much time.
No problem. Thank you.
Okay, so then the third aspect I want to talk about is maybe is more this intervention-induced MRI changes. It's more to disentangle what is now, which changes during primary school are now more maturational changes and which are more reading-induced. So we want to see, can learning to read really change the brain? So we will investigate that mainly during the more plastic or we assume that's during the more plastic stages of reading development, so more at the early stages of reading developments. So if we look at previous studies on intervention-induced MRI changes in the domain of reading, we see there's 2 diffusion studies that looked at the effect of reading intervention on the white matter. And so for example, in the study of Keller, they found a region where children poor readers who received intervention, they showed a larger increase in FA than children, poor readers again, that did not receive intervention. And also in the study of Huber, they also find more widespread changes due to intervention. But this was all in school-age children. And also they never had a well-controlled, they did not have a group that followed also a control intervention, so that was actively involved, but not in reading, but maybe in another type of intervention. So therefore, it's sometimes a bit difficult to see what is placebo effects, what is maturational effects. And also, it did not include the pre-reading stage, whereas we are convinced that also based on our data of the gray matter, that the reading network is probably more plastic at an early stage, so we should intervene at an early stage. So this was the design of the next study, which is part of the PhD of Maria Economou, and also of, so she's working on the MRI data, and Femke van den Bent is working on the behavioral data. So I will present some of these data. There's also a third PhD student working on the EEG data, but I will not present that today. So, so the design of that study is to look mainly within this early stage of reading, so even before reading onset, so in kindergarten, we have MRI scans, and we also have 1 MRI scan at the end of grade 1. I will not discuss these data because they were just very recently acquired, but I will mainly focus on the scan just prior to the intervention and directly after. And the intervention was a tablet-based intervention that the children did for 12 weeks. And they trained for 6 days per week around 50 minutes of training. And so we had one group. Yeah, so maybe first important to say that all of these children they had a risk for dyslexia. So meaning that based on a cognitive screening, they had low scores of phonological awareness, rapid automatic naming, and letter knowledge. And we saw that, and we divided them in a group that received graphogame intervention, so which is a literacy intervention. We adjusted this intervention to pre-readers so that it included a lot of letter knowledge tasks, but also some phonological awareness tasks. And the other pre-readers at risk, they received an active control intervention. So they played some Lego games. And then we also added a group which was not at risk. So they were just typically developing children, no increased risk for dyslexia. And they also did not receive any intervention, just as a, just to see what the typical development would be. The study also includes another type of intervention with another intervention group of at-risk pre-readers, but I will not discuss that today. I will focus on the intervention of the literacy, so on the graphogame intervention. So what did we see now first behaviorally? So we see that it has the graphogame has the effect that we wanted it to have, in the sense that you see that letter knowledge is increasing in the graphogame group. So from pre to post, as you see an increase here in letter knowledge. You don't see that in the active control group. They stay rather stable. And also in the non-risk control group, they did not receive an intervention, but that were also not at risk. It's rather stable. And of course, you see that they are performing, the non-risk group performs better on letter knowledge than the two at-risk groups, because this is the way they were selected. Okay, so that's the behavioral data, but that's not very in detail, because I want to focus on the brain data. With regard to the white matter, what did we see? So first of all, with regard to the method, we again used the automated fiber quantification. We looked at the long segment of the arcuate fasciculus and at the IFOF, and also at the anterior segment of the arcuate fasciculus, or which is called in AFQ more the SLF, so which is sometimes a bit confusing, but just important to know is that at the moment we have the plots, but it's not yet statistically analyzed, because Maria is working on a lot of very fancy statistical models, like the general additive models, which can take into account that you have all these nodes clustered within a tract of one subject, but it's also a non-linear modeling that is needed. So, but at the moment we just have the plot, so it's just a visual inspection. If we look at the left arcuate fasciculus, we do see that, so this is the pre-test in green, and then the post-test is in orange. We do see in the graphogame group that there seems to be a slight increase at these two peaks, probably then due to the intervention, because we don't see that very much in the active control group or in the non-risk children. But of course, it's very preliminary, and we don't know yet whether this is whether it's also a statistical effect. We did not see a lot of these things in the other tracts, except for in the right as a left, so which is also the tract that was indicated in previous studies by the group of GAP as a tract that could maybe be a protective mechanism against reading problems. We do see that the graphogame group seems to increase also there in the FA of this right as a left, whereas the other two groups don't really show this increase. But of course, as I said, very preliminary data. If we look at the gray matter, again, it's preliminary data, but at least we have, we did some free surfer analysis. So again, the cross-sectional pipeline for the moment. And we looked at these reading regions that we also selected in our previous longitudinal study. Again, here, as I said, it's still preliminary. We will analyze the data with the longitudinal free surfer data, because now we have the two waves, so we can do the longitudinal pipeline. In contrast to the previous study, the longitudinal study, where we had changes across whole primary school, and where the growth, where there was a lot of growth in the head size, we have now less that problem, because the the time point, the difference between the first and the second time point is only 3 months. But this is something to keep in mind that at the moment it's still analyzed cross-sectional, so it's a bit less reliable probably than the longitudinal pipeline. But so what did we see? If we looked at statistics, we did not see a three-way interaction, so with regions, so it's not, but we did see a two-way interaction between group and time. So meaning that the graphogame group was increasing more in gray matter than the other two groups. But this effect, so this increase specific for the graphogame group, was not specific to a certain region that we included. So we included the 6 regions in the left, being part of the reading network, but also the ones in the right. So we could not really indicate whether it was in certain regions more than in others. It seems to be some kind of overall effect. So what can we conclude? At the moment, not so much, because many more analyses are needed. But there is some evidence, maybe at least on the visual inspection of the diffusion data and the early statistics of the gray matter data, that the literacy intervention, so the graphogame, is leading to some brain plasticity, structural brain plasticity in these pre-reading children who are at risk for dyslexia. So that's a bit about this third pillar. And then finally, I will say something about this, the more intergenerational imaging, because of course, parents, they influence children's reading ability. They do that through the environment they provide, but also by the genes they transfer. So in that perspective, it's also interesting to see how the brain of a child is that similar to the brain of the parent, and can that also help in explaining the reading development of the child. If we look a bit to existing literature, I think a good way is to start from the multiple deficit model. So which is if you focus on the child, which is what you see here in purple, so the reading problems the child can have, it's determined by a lot of factors at different levels. So at the cognitive level, at the neural level, and genetic level, and all these factors interactively, there's dynamic interactions between these factors, and at the end, this determines whether or not you develop reading problems. But what's important in this intergenerational multiple deficit model is that there's an extra layer for the parents, and meaning that all these different levels of the child is influenced by what the parents provide, by the environment they provide, but also by the genes they provide. And this, in that perspective, there were a lot of studies also done on looking at correlations between parents' reading and children's reading. So they find some similarities, but it's at the moment not yet understood how these underlying mechanisms, how do they explain this relation between parents' reading and children's reading. We know from some studies in twins, for example, that probably the role of the environment is rather small in these intergenerational correlations for reading, that it's more driven by genetic or biological factors. We also know from large-scale diffusion studies that the FA in the white matter tracts, that it's highly heritable. So in that perspective, we wanted to see, can we see similarities between the white matter tract of the parents and the white matter tract of the child? And can this help in explaining the reading development of a child? So for that study, so that was part of my postdoc stay at UCSF with Fumiko. So there was a data set available in 33 families. So we had diffusion data of children in grade 3. We also had diffusion data of their father and of their mother. And then we investigated through manual tracing again of these tracts in the children and in the parents. We looked at the FA in bilateral arcuate fasciculus and bilateral IFOF. We first examined the relation between the parents' FA in the white matter tracts and the children FA, and we saw that mainly in the bilateral IFOF, we saw some correlations between the tract of the father and the mother on the one hand, and the one of the child on the other hand. We also saw that that parental IFOF, that explains a lot of the relation between the children's IFOF and the reading development. So a lot of this relationship is explained by parental IFOF. We did not see any intergenerational correlations for the Arcuate fasciculus, which was maybe a bit unexpected, because also in diffusion studies, often there is high heritability rates for the Arcuate fasciculus. But on the other hand, it could maybe fit with the idea that the IFOF is a tract that matures earlier in development. And so maybe more determined by the parents, by the biological transmission of the parents. And maybe the Arcuate fasciculus is still developing throughout the first years of life. And maybe there is more influence of the home or language environment they grow up in. So this is something, of course, that needs further investigation. So in general, the conclusions with regard to the pre-reading MRI markers of dyslexia, we see some early deficits in the reading network. So in this long Arcuate fasciculus and the left fusiform, although we couldn't really say it's specific to the left, because we also saw some deficits in the right, which is different than what we observed in adults. With regard to the changes, the neural changes in the structure of the white matter and the gray matter, we first of all that white matter throughout primary school has more like a linear increase, whereas gray matter there it's very much dependent on which region you investigate and also which reading stage you investigate. So it's different for the first years of primary school than for the later years of primary school. And also with regard to this functional specialization, dorsal sustaining more phonological aspects and the ventral tracts sustaining more orthographic aspects, we see this is something that gradually emerges throughout the first years of reading acquisition. So by grade 2, it's there, but it was not yet there in pre-reading school. With regard to dyslexia, we did not find a lot of evidence for a different neural trajectory within the left reading network. We did see some differences in right regions. With regard to the intervention, so the very early intervention in the pre-readers who were at risk for dyslexia, of course, we don't have any final results yet, but there seems to be some plasticity which is induced by this intervention, the reading intervention. And with regard to this intergenerational imaging, we see mainly correspondence between parents and children's IFOF, not so much in the Arcuate fasciculus, but this might be due to more environmental influences early in life on the Arcuate fasciculus and maybe less on the IFOF. So I think this is the main summary of my talk today. So I want to thank you all for listening, and I'm very happy to answer your question or to listen to your feedback. Thank you.
I am going to unmute everyone in case people could thank the speaker for the wonderful talk. Thank you, Maaike. That was great. I always wanted to get a full hour and hear a lot of everything, a little bit of everything in there. So it's great instead of hearing shorter talks that I've heard from you. So that's fantastic. Thank you very much. I know some people are needing to leave because it's an hour, but we did allocate more than an hour for us to have some Q&A questions. Would anyone like to ask questions? Feel free to unmute yourself and ask directly rather than typing it in if it's easier. I have a bunch of questions, but I'm going to hold mine to the end unless we run out of questions. But anyone? Simple clarification question to start with. How do you identify family history or a certain that in your sample, and how does it compare to other groups doing it?
Yeah, I think, now we did a lot of group comparisons. So then we classified children with a family risk versus non-family risk, and this was based on whether they had a first-degree relative with a family risk. But we do have also, not in all of the parents, but many of them filled also in the adult reading history questionnaire. So this allows us to look a bit more as at the family risk as a continuum. So not just dividing them in with and without family risk, but more in looking at how the family risk as a continuum can relate to the reading skills. So in some of the studies, we use also this family risk in a continuous way, but in most of the things that I presented, it was more like the group comparison, because this is how they were originally recruited.
And so it's based on a formal clinician's diagnosis or psychologist piece of paper they come in that says you have had dyslexia when you were a child, sort of.
Yeah, indeed. But it's not always the parent, so it can also be a sibling. So I think that's maybe sometimes different than other studies. So they had to have an official document with a diagnosis, but it was of the first degree relative. So it could be a parent, but also a sibling.
Great. Other questions? I do want to be mindful of everyone's time, especially in Europe when it's running into the late evening and a lot of people have children. But can I ask one more question? And feel free, if you have questions, to interrupt me and veto me, because I'm going to end up monopolizing Maaike's time, but where we could set up a different time. But if you look at some of the older behavioral literature and look at the genetic transmission or heritability being higher for phonological processing and environmental being orthographic processing more environmentally mediated, relatively speaking. And I think there's some papers that allude to this or conclude so. Does that seem to contradict with some of your findings that maybe the AF seems to be almost showing the opposite with the ventral dorsal kind of findings?
Yeah, I think indeed, based on these studies, if phonological skills are more heritable or more genetically driven than orthographic skills, then you would assume more intergenerational correlations in the arcuate fasciculus more than in the ventral IFOF, but which was not what we found. We found then more the opposite pattern. Yeah, part of it can be due to the fact that maybe in these young children, you don't have this clear functional distinction yet between dorsal and ventral. So maybe in these younger children, you have, as I also show with the pre-reading data, you might have for phonological skills also ventral regions to be involved. So it's maybe based on that not so clear whether you can expect in the young children also more genetic transfer in the dorsal regions and less in the ventral, because you're not sure whether the neural underpinnings for these skills are the same in adults as in children. Yeah.
And then you said that, sorry for interrupting, but you said that because the dorsal might be developing more. So was that relationship in relation to saying, so is that the same thing as you saying, for example, in adults, so the adults AF might look quite different from the children, even though functionally it might be more genetically transmitted if you look at the cognitive process. So then it's more of a function of somehow finding a way to take into account these developmental deviations? Or you were talking about it more from, I think you were talking about it more different way.
Yeah, it was more based on developmental studies that often show that this ventral tract is more developed from a very early age, whereas you see more changes in the arcuate fasciculus also at later stages. And there was, for example, also a recent study where they showed that, for example, the amount of language exposure during the very first years of life, that this has an impact on the arcuate fasciculus also at a later stage. So there was even in adolescence, you still could see that relationship. So yeah, for me, it seems to suggest a bit that maybe this arcuate fasciculus is more prone to environmental induced changes. So also with regard to the intervention study, I would also expect mainly changes in this arcuate fasciculus more than in the IFOF, because this IFOF is normally already more developed at an early age. but it's a lot of speculation, and it's, of course, we need more studies to verify this.
Yeah, it almost seems like also it could be just methodological kind of MRI constraint sort of artifact, possibly also because the association areas and temporal parietal is more variable across individuals just in general, and then it's more of the later evolved during evolution, but also during development also, as you say, and so that might be an artifact of that. So it seems like there's some way, I don't want to call it artifact because it's a true effect, but harder to capture these kind of intergenerational transmission, even if it's more heritable or whatever it might be.
I agree. Yeah, indeed.
I have more questions, but I'm going to let other people ask. And if not, feel free to rotate, get off the call if you need to go, and don't feel like you need to be polite, because I'm going to monopolize her with one more question, if that's okay. Because I'm sure some people who are staying on might be interested, because I get confused, and I asked you so many times, even though it's the same data, it's the data set that we worked on. But so typically when I think about intergenerational transmission and look at the kind of the mediation model, you would think you have the parent brain and then the child's behavior, and then, or a child's brain and child's behavior as a way of hierarchy, but you have it as the in-between in the triangle at the top. And you explained to me a number of times why, and it might be driven more by the results, but your interpretation wise and how you link that to potential protective factors. Do you mind? I'm sure some people would be curious to hear.
Yeah, I think we started from the observation there was some relationships between children's white matter tracts and their reading scores, especially reading development. So it seems to be, at least in the group we had here, it was all children, or at least there was a proportion of them had a risk for dyslexia, but at the end, none of them developed dyslexia. So I think, and we saw mainly that the degree by which they increased in reading skills, that's related to the right tracts, such as the right IFF, for example, but also the right adcord fasciculus. So it could mean that the more that this is some kind of compensation again of the right, so that although they were at risk, eventually they did not develop reading problems, but they developed quite good reading skills. And then the question is, so we see that relationship between the right tracts and the reading development. So then the question is this relationship explained by the parents' biology? So, and that seems to be the case, because that relation within the child, between the child's brain and behavioral, is disappearing when you include the parental brain. So I think it shows a bit that the brain of the parent is explaining a lot of that relationship. So that that a lot of the variance can be explained by the parents. So it means that the brain of the parents is partially transferred to the child, and that is explaining a lot of the relationship with reading development.
Got it. Thank you very much. Anyone else have questions before we close? All right. Thank you, everyone, for letting me monopolize Maaike and ask these questions in person. Well, thank you very much for everyone for joining us. Thank you, Maaike, for the wonderful talk. I think I see a lot of in the